a master's in psychology, neuropsychology concentration. As promised, I want to finish reading this article. This is exposure to 835 megahertz radio frequency electromagnetic field decreases the expression of calcium channels, inhibits apoptosis, but induces autophagy in the mouse hippocampus. This was written by Ju Hwan Kim and Yu Dong Son and Hak Rim Kim. So in the last video, I only got up to, I believe it was just about after the introduction. So the introduction basically talks about the adverse effects that have been noted by the World Health Organization. The International Agency for Research on Cancer classified RF-EMFs from cell phones as a possible human carcinogen, class 2B. Many scientists have recognized radio frequency electromagnetic fields as a potential cause to serious adverse risks to human health. The main concern is the central nervous system due to the close proximity between mobile phones and the brain during communication. Research on radio frequency radiation has demonstrated a negative impact on neurological factors such as neurodevelopment, the blood-brain barrier, neurite outgrowth, neurotransmitter release, cognitive impairment, and ultimately behavior. Additionally, additional cellular effects of radio frequency electromagnetic fields exposure included alterations of intracellular and molecular pathways, such as the extracellular signal-regulated kinase pathway, apoptosis, and regulation of the cell cycle. In addition, autophagy was induced in mammalian cells after exposures of 835 megahertz and 1800 megahertz radio frequency electromagnetic fields. Autophagy is a destructive mechanism in which unnecessary proteins or damaged organelles are encapsulated by an autophagosome, which then fuses with the lysosome. So basically it encapsulates it, and it says that that's part of the homeostatic process to rid the cellular environment of those basically damaged cells, but damaged parts of cells, organelles, proteins. When the mice brains, or brains in any way, human brains, rabbit brains, are being radiated at that specific frequency, and it goes into the power level as well in this specific study. The hippocampus belongs to the limbic system and plays a crucial role in the formation of new memories and spatial navigation, and is also related to learning and emotions. Importantly, dysfunction of the hippocampus may lead to early Alzheimer's disease. So basically it talks about shrinking hippocampus neurons, is involved in severe mental disorders like schizophrenia and major depressive disorders. Exposure to radio frequency electromagnetic fields leads to impairment of intracellular calcium homeostasis in the murine hippocampus. So basically exposure to the radio frequency electromagnetic fields harms the hippocampus, the memory and learning centers of the brain. It says alterations, that happens through alterations in the intracellular environment, the calcium concentrations. Because of those alterations, it can stimulate a variety of pathogenic events. So pathology is no good. It's basically disease or harming, or it could even be killing the cells in that instance. It includes apoptosis, which is cell death. That's basically from changing the calcium concentrations, alterations inside the intracellular calcium concentrations. It goes on to talk about the calcium influx, the voltage-gated calcium channels, is an important factor for the initial steps of apoptosis through the regulation of calcium ion entry across the plasma membrane. So those channels that are affected during the electromagnetic field radio frequency experience a change such that those gates are now open and more calcium can get into the cell than is usually needed, or it can be the actual cell could experience calcium overload. A high level of calcium in mitochondria is essential to trigger apoptosis. So once a certain amount of calcium gets in to the energy center of the neurons in the brain, then the cell dies. So that's how it works. It does talk about some positive effects that were seen after 28 days of these mice, which were studied in a lab in Korea. They were radiated for 28 days, radio frequency electromagnetic field radiation using an RF radio frequency generator, and it basically attempted to match the same amount of radiation that people experience or that is emitted from telecommunication cell phone towers all across many different countries. So that's 829 through 839 megahertz. 829 through 839 megahertz. The middle point is 835. They're considered the main frequencies and are actively used by telecommunications companies. This article was published in 2018. It was written in 2017. The everyday use and predominant exposure of 835 megahertz radio frequency electromagnetic fields has made the finding of any biological effects to be of paramount importance. So the everyday use of those ones that we experience every day has made the finding of any biological effects to be of paramount importance, because if we use something every day, we want to know what the effects are, no matter what it is. So they studied the whole body exposure was at a specific absorption rate, which is called SAR value, SAR, of 4.0 watts per kilogram of body weight for 5 hours a day and for 4 weeks. I don't know who was actually on the phone for 4 hours, 5 hours a day with the phone that close to them every single day, but I put up read articles from amazing researchers that have shown that there's links to brain cancers that were found because of continued cell phone usage. Now, it's not to scare everybody because most people don't experience that, but those that do experience that, they report it to their doctors, whatever country that they're in, and then those places that actually study that, they're the ones who put that up so that other people know what could, be harming them or their children, or even the elderly. And that's basically the science in 2011 that ramped up the cell phone radiation from possible for up to what it is currently a class 2B carcinogen, so meaning possibly carcinogenic. The next level up, I believe, is possible human carcinogen. That's class 2B is where cell phone radiation is at currently. So it was brought up one level in 2000 because of this study that was put up in 2011. The next step is, I think, likely or somewhat likely, and then it goes up in level until, it says, basically it causes cancer. And that's based on any evidence, and that's based on the way that they study it is important. And the models that they use in studying it are also important. Are those models actually indicative of real life experience every day? Do they match up with what kids are using every single day? Do they match up with any of these other what people use every single day? How did they measure it? What did they use to measure it? Those types of things. And they get better with that because people point out what the mistakes are, particularly what the benefits are, what the cons are, what the mistakes are in studying it, how to get better and more accurate at studying it. If the person's a kid, then they have bigger heads and more water, which means that the thermal effects can be increased in their children. So that's one explanation as to why kids may be more affected by cell phone radiation from cell phone towers than adults. But if they're a kid, they got to stay alive until they're an adult to make that safe category. Just like when you're driving and you're 18 years old or 16, you get your permit license, you're at the highest risk of getting into an accident or dying. Then you get to 30 and you're no longer at that risk, but you have to live to make it to that safer likelihood, right? So that's the way I understand it. It says the whole body exposure was at a specific absorption rate of 4.0 watts per kilogram for 5 hours a day for 4 weeks. Additionally, in the safety level, 4.0 watts per kilogram specific absorption rate value is the maximum permitted exposure to human limbs, but not whole body. But the whole body is allowed up to 0.08 watts per kilogram of specific absorption rate. So throughout the whole body, it has to be a lot lower, right? So if it's a certain level at the limbs, that's just the limbs. But throughout the whole body, it has to be much lower. I don't know why that's like that. Maybe the limbs have some type of, they can withstand more, maybe. I don't know, but that's what it is. So throughout the whole body, they can only absorb so much, and that's for the general public person in accordance with the International Commission on Non-Ionizing Radiation Protection, the ICNIRP guidelines. Some other points and research points that were brought up were the fact that much of the research has looked into thermal effects, but there are also non-thermal effects. So it's not just the heat; it has to do with what might be occurring. The cumulative effects of non-thermal radiation from this. So first they look at, is it heating them too much? Then they say, well, there are some effects, but now how do we investigate? What do we look at if there's micro changes in DNA? How do you know what to do if you don't if that's not pointed out as something that could also likely be occurring? So they're also studying non-thermal effects as well. The SAR generated by our RF generator was evaluated by measuring E field at the phantom position in the air, while simultaneously by considering the ratio of E field in the liquid to E field in in the air at the same position in the same environment. Following the calculation of the SAR value, the value is 4.14 watts per kilogram, which is proximity of setting up out power at SAR value of 4.0 watts per kilogram of horn antenna of the exposure apparatus. As a result, measurement of radio frequency signal and SAR value generated from our radio frequency EMF generator produces 835 megahertz of radio frequency EMF with 4.0 watts per kilogram SAR value. More information about the exposure system has been described in details in our previous reports. The mice in control group received sham exposure in parallel for 4 weeks. Sham exposure group was maintained under the same environmental conditions except for the exposure of radio frequency electromagnetic field radio frequency radiation. Both groups were treated in the same circular pattern from housing cage to exposure cage. Throughout the experiment, the mice could move freely and were not restricted to access food and water during the exposure. The cage for radio frequency electromagnetic field exposure was placed in the RF-EMF generator. 835 megahertz megahertz. Okay. 835 megahertz radio frequency electromagnetic fields was delivered to the mice from a horn antenna installed above the mouse cage. The bottom and walls of the cage were covered by ceramic wave absorption material. All the animal experiments were completed under an automatic lighting system and air conditioning facility, which was maintained in constant temperature. Quantitative real-time PCR and semi-quantitative real-time PCR. In order to determine the autophagy is also triggered in the hippocampus of mice brains following the 4-week exposure to the EMFs, the radio frequency electromagnetic fields. The selected autophagy genes such as ATG4A, B/B, and ATG5, ATG9A, Backlin1-/2, and LC3AB were examined. The expression levels of these genes were found by using either quantitative real-time PCR or semi-quantitative real-time PCR, and changes were recorded. Mice were euthanized the day following the completion of the radio frequency electromagnetic field exposure for 4 weeks. After the mouse was euthanized by cervical dislocation and head was decapitated with scissors, the whole hippocampus was isolated from the brain. The dissected hippocampus was immediately frozen and stored at -80 degrees Celsius. Total RNA was extracted from the whole hippocampus by using trizol reagent. That's from Thermo Fisher Scientific USA. Purified RNA was reverse transcribed to cDNA using MMLV reverse transcriptase, Bioneer, South Korea, and an oligo-D T18 primer. Quantitative RT-PCR reactions were carried out with rotor gene SYBR green supermix kit QIA Gen Germany, and fluorescence was measured using the rotor gene PCR cycler from QIA Gen Company. The expression levels of the genes were normalized to that of GAPDH as a housekeeping gene. GAPDH primer was purchased from QIA Gen Germany. The primers used for the quantitative real-time PCR and semi-quantitative real-time PCR Table 1 were synthesized from Bioneer or Cosmogen Tech, South Korea. The 5 biologically independently prepared samples by pooling were tested, and each PCR reaction was done in triplicate. The relative levels of the specific mRNA were calculated by normalizing the expression of GAPDH by 2 superscript negative delta delta CT method, n is equal to 10. Also, the expression values of the RF, radio frequency electromagnetic field exposed groups, were normalized to those of the sham exposed group. Semi-quantitative RT-PCR reactions were performed using PCR premix Bioneer South Korea. Subsequently, semi-quantitative PCR product of genes was electrophoresed in 1.5% agarose gel, and signal intensity of PCR product was visualized the Cyto60 Lee Core USA stained DNA using the Odyssey infrared imaging system, Lee Core USA. Sequences of oligonucleotide used for RT, that's real-time PCR. Western blot analysis. The hippocampus was quickly dissected from brain of mice and one side of hippocampus from each mouse. N is equal to 10 was quickly lysed with RIPA lysis buffer ATTO Japan, which was supplemented with protease inhibitor and phosphate inhibitor cocktail ATTO Japan. The hippocampal lysates were then homogenized and sonicated briefly in ice. For Western blot, pooling samples were used, and depending on protein concentration was determined by Lowry protein assay, Biorad USA. Lysated samples were separated by electrophoresis on 10 to 15% of SDS polyacrylamide gel and transferred to polyvinylidene difluoride, PVDF membrane, ATTO Japan. Membranes were blocked in Odyssey blocking buffer, Lee Core, Lincoln, Nebraska, USA for an hour at room temperature, then incubated in a specific primary antibody solution. Primary antibodies, ABS, were used. One anti-LC3B, AB, that's from Cell Signaling Technology USA. Number 2 antibody that was used was anti-Eklund-2, AB. Cell signaling also, it's also from Cell Signaling Technology USA. Number 3 was anti-BC12AB. That's for the other antibody. That's from Santa Cruz Biotechnology USA. Number 4 was anti-Bax AB, Santa Cruz Biotechnology USA. And 5, anti-Pan calcium channel AB, Sigma Aldrich USA, binding all types of alpha 1 subunits of voltage-gated calcium channels. The GOAT anti-rabbit 800CW or GOAT anti-mouse 680RD from Lee Core USA were used as secondary antibodies. The membrane was washed thoroughly with tris-buffed saline containing 0.1% 20 micromoles, tris 137 micromoles. Sodium chloride was 0.1% 20, pH was 7.4. Protein bands were quantified by using Odyssey infrared imaging system, Lee Core USA, and normalized by using a tubulin band. Transmission electron microscopy, TEM, transmission electron microscopy. The dissected hippocampus were fixed immediately in 2% glutaraldehyde, 2% paraformaldehyde in a 0.1 molar phosphate buffer. That's the pH was 7.4 for that. For 2 hours that occurred at 4 degrees Celsius. Following 3 washes in the phosphate buffer, the brain tissues were post-fixed with 1% osmium tetroxide on ice for 2 hours and washed 3 times, all in phosphate buffer. The tissues were then embedded in EPON 812 mixture after dehydration in an ethanol and propylene oxide series. Polymerization was conducted with pure resin at 70 degrees Celsius for 24 hours. Ultra thin sections at 70 nanometers were obtained with ultra microtome, that's MT-X, RMC, Tucson, Arizona, USA, and then collected on 100 mesh copper grids. After staining with 2% uranyl acetate 15 minutes and lead citrate for 5 minutes, the sections were visualized using the transmission electron microscopy, TEM. That's from Techni G Squared Spirit Twin, FEI, Hillsboro, Oregon, USA, at 120 kilovolts. The whole pair of hippocampus was used for the TEM study. That was 3, n is equal to 3. Statistical analysis. All data was presented as the mean of the SEM. The significances of all data assessed by two-tailed student t-test with probability values of less than p is less than 0.05, so 95% confidence interval was statistically significant. We used GraphPad Prism 4 program, GraphPad Software Inc., La Jolla, California, for statistical analysis. Results: decreased expression of voltage-gated calcium channels in the hippocampus. To investigate the effects of radio frequency electromagnetic field exposure on calcium homeostasis in the hippocampus, we focused on voltage-gated calcium channels, which controls intracellular calcium concentrations. Total protein extracted from the hippocampus was used to perform Western blots using an anti-pan calcium antibody. That's from Sigma, St. Louis, USA, which recognizes all types of alpha-1 subunits of voltage-gated calcium channels. And the anti-pan calcium channel antibody. All subtypes, alpha-1, all subunits. The data showed that 4-week exposure to radio frequency electromagnetic fields decreased the expression of voltage-gated calcium channels in the mouse hippocampus. These data strongly suggested that the exposure to radio frequency electromagnetic fields could induce alterations to calcium homeostasis in the mouse hippocampus. So all that just means is that it disturbs the normal amounts of it just disturbs the normal amounts or the typical amounts of calcium that is in the brain and the neurons. That's all that means. It disturbs the homeostatic rhythm of that inside the mouse's hippocampus. Has a picture here and a figure. Sorry if I'm looking around. I'm in a parking lot before I start my work, so I want to make sure I'm aware of my surroundings. The expression level of BAX is down, but that of BC12 is up in the hippocampus after radio frequency electromagnetic field exposure. To elucidate the effects of 4-week radio frequency EMF exposure on apoptosis, that's cell death, the anti-apoptotic factor BC12 and the pro-apoptotic factor BAX were analyzed in the hippocampus with both RT-PCR, and RT is the real time, and the other one is the semi-quantitative. So RT-PCR and Western blotting. The real-time PCR results revealed that the transcript level of the pro-apoptotic gene BAX slightly declined in the hippocampus of the RF-EMF exposed mice. That's in figure 2. So there was a little bit less of that gene that was expressed inside the mouse hippocampus that actually kills the cells at that level of electromagnetic field frequency, at that frequency and that power level for that specific amount of days. By nearly, it says, similarly, immunoblotting analysis indicated that the level of anti-apoptotic BC12 protein was significantly increased, okay, while the VAX protein expression was significantly decreased. These results indicated that the apoptotic pathway may be hindered following a 4-week exposure of radiofrequency electromagnetic field exposure. Autophagy-related genes are significantly upregulated in the hippocampus following radiofrequency electromagnetic field exposure. To determine whether autophagy is triggered in the mouse hippocampus following 4-week exposure of radiofrequency electromagnetic fields, we examined the expression of selected autophagy genes. That's the ones that I read before, the ATG4A/B, the ATG5, the ATG9A, the Becklin1/2, and the LC3AB using the quantitative real-time PCR, QRT-PCR, and semi-quantitative RT-PCR. That's the SQRT-PCR. So as shown in the figure 3, the QRT-PCR results indicated that the expression of most of the genes related to autophagy, the ATG4B, ATG5, ATG9A, Becklin2, and LC3B, were significantly increased at about 1.5 to 2-fold as compared to the controls in the hippocampus. However, the expression of the ATG4A, Becklin, did not differ from that in the control animals following the RF-EMF exposure. So the expression of that gene, of that specific gene, ATG4A, and the expression of ATG4A, Becklin1, did not differ. So between those mouse that were experienced the radio frequency electromagnetic fields and those that did, there was no difference between those in those specific autophagy genes. So 4B up, ATG4A stayed the same, Becklin1 stayed the same, Becklin2 up, ATG5 up, ATG9A up, LC3B all significantly raised, but 2 of them stayed the same. In addition, SQRT-PCR was carried out to further validate the changes in gene expression found in quantitative real-time PCR. Generally, the results from SQ-PCR were consistent with the patterns seen with the QRT-PCR data, indicating that ATG4A, ATG5, ATG9A, and Becklin2, and LC3A/B showed higher intensity in the radiofrequency electromagnetic field group compared to controls. Analysis of the QRT-PCR indicated that there is upregulation of genes related to autophagy in the hippocampus in response to 4-week exposure to radio frequency electromagnetic fields. Autophagy structures are accumulated in the hippocampus. In parallel with the upregulation of mRNA, that's messenger RNA, level of autophagy in the hippocampus depending on radio frequency electromagnetic field exposure, we further explore the levels of autophagy-related genes, proteins, such as LC3B2 and Beclin-1, which play a key role in the formation of the autophagosome. Sorry, the phone cut off. We were interrupted there. Just a side note, if you ever believe that something is not right, never be afraid of investigating that, of complaining about that, and looking into that for yourself. No matter who is against that, if it's important to you, it may be important to someone else, and if it's the right thing to do and you feel that it is such, anybody can be wrong, but the way to look that up, to figure that out, is to actually look into it. So I'm African American, and many times things such as the Tuskegee experiment come to mind. People that have been through pretty much everything, and yeah, it doesn't hurt to check and to complain and to file a petition and to state what occurs, because it's your life. It's no one else's life. And nobody can tell you what how best to fulfill that. That's the way that you do that, by asking questions, by complaining, by looking into things, so that eventually everybody has a better life. And then those people that chose not to, that was their choice. Autophagy structures are accumulated in the hippocampus. So, with the upregulation of the messenger RNA level of the autophagy in the hippocampus, depending on the radio frequency exposure, they further explore the autophagy-related proteins, such as LC3B2 and Beclin-1, which play a key role in the formation of autophagosome in response, or phagosome in response to environmental cellular stress. So the only reason that the autophagosome actually comes online, right, is because of environmental, or actually, to say it more accurately, is because of environmental cellular stress. Not only, but because of. The LC3B-2 protein level was, and I just got some random comment and link on there from somebody who's currently watching on this stream. The LC3B2 protein level was significantly increased by 1.7-fold compared to control in the hippocampus, but the expression of Beclin-1 was not changed. Ultra structural comparisons between sham and radio frequency electromagnetic field exposed hippocampal neurons was conducted using TEM. That's the microscope. The results indicated that the augmented number of autophagy structures, such as autophagosomes and autolysosomes, were observed in hippocampal neurons following radio frequency electromagnetic field exposure. That's in figure 4B. These results indicated that 4-week exposure to radiofrequency electromagnetic fields could induce higher LC3B2 expression and accumulate more autophagy structures in comparison to the sham group in their hippocampuses. So you see a change in those autophagy structures due to the radiation or the radio frequency electromagnetic field exposure at 835 megahertz and 4 watts per kilogram of body weight. The data for 4 weeks. The data strongly suggested the activation of the autophagy pathways triggered in the mouse hippocampus by radiofrequency electromagnetic field exposure. Discussion. This study evaluated the effects of radiofrequency electromagnetic field exposure on calcium channel expression. Intracellular calcium concentrations are regulated by calcium channels and affect various neuronal processes. Exposure to radiofrequency electromagnetic fields caused impairment of intracellular calcium homeostasis in the murine hippocampus. The calcium ion is an essential component for the regulation of a variety of cellular processes, including apoptosis. The impairment of the calcium homeostasis in a cell may lead to activation of apoptosis. In this study, we showed that exposure to radiofrequency electromagnetic fields may alter the calcium homeostasis in the hippocampus of mice by decreasing the level of pan-calcium channels, which regulate calcium influx. So they downregulate. There's not as many of the calcium channels for the calcium to connect to those neurotransmitters. Calcium homeostasis can be regulated by several types of calcium channels, including voltage-gated calcium channels. Voltage-gated calcium channels are responsible for fast calcium influx into the cell, which controls the entry of calcium ions across the plasma membrane. Intracellular calcium overload may activate pro-apoptotic genes. So if it gets overloaded with calcium, the cell, that neuron that is, then apoptosis genes start to pop up. And then the apoptosis process could follow after that. So it says here, those activate pro-apoptotic genes due to the intracellular calcium overload, and that which causes degeneration of neurons. That's neurons are in the brain. It's the cells in the brain. And ultimately cell death. It has also been suggested that capacitive calcium influx through calcium release activated calcium channels causes apoptosis. So there's also calcium release activated calcium channels that also may have to do with this apoptosis. However, the inhibition of calcium channels in neurons, which causes a reduction in the level of intracellular calcium, so inhibition of the calcium channel neurons, channels in neurons, which causes a reduction in the level of intracellular calcium, may serve as a neuroprotective function to promote neuronal survival. So basically, at that level of 835 megahertz, there's less of those channels, calcium channels, for the neuron, the neurotransmitters to receive that calcium that is abundant in the synaptic cleft between the neuron and the dendrite, which sends the signals across the brain, the nerves in the brain. So if there's less of those shipping ports, I'd say, right, there's only 2 shipping, 3 instead of 4, then there's a lot less of those neurotransmitters to connect and move into the cell, which they're stating serves a protective function. So there's not an overload of calcium inside the neuron, basically overloading if the calcium gets to a certain point to where it overloads the mitochondria, the energy center of the brain, the energy center of the cell, the neuron, then that's where the neuron becomes damaged and apoptosis can occur. So in this research study, it basically showed a benefit to those mice. It says, in the present study, we found significant augmentation of both autophagy-related genes and proteins in the hippocampus following a 4-week exposure of 835 megahertz radio frequency electromagnetic field exposure with 4.0 watts per kilogram of specific absorption rate, or SAR value. Several studies previously suggested that intracellular calcium is an important regulator for initiation of autophagy processes. Additionally, the autophagic flux was reduced by increased cytosolic calcium concentration. So, due to prevent the fusion between autophagosomes or phagosomes and lysosomes in hepatocytes. Prevention of cytosolic calcium influx by calcium channel blockers effectively restored autophagic flux, presenting an accumulation of autophagosomes and increase in LC3-2 level in the obesity-induced autophagy arrest model. So in that specific research example, calcium channel blockers effectively restored the change in the autophagy that was occurring. Right, so that would be the autophagosomes and autophagosomes and the lysosomes. So it says that prevention of the cytosolic calcium influx by calcium channel blockers effectively restore the autophagic flux, presenting by accumulation of autophagosomes or phagosomes and increase in LC3-2 level in the obesity-induced autophagy arrest model. As calcium channel blockers can prevent cytosolic calcium influx, inhibition of calcium channels could be a molecular mechanism for initiation of autophagic processes in autophagy defects. Right, so it seems that it's saying here that for organisms that, or I'm assuming they're talking about people that have defects in their autophagic response, then they could use calcium channel blockers. Right, so that would be to prevent the cytosolic calcium influx. So it's saying that those calcium channels could basically protect those individuals who do not have autophagic processes because their autophagy is defective, right? It's not working correctly. Once again, this is for information purposes only. I'm not stating that anybody should or could do anything. You have to ask your doctor if anything is safe for you, what the side effects could be, if they're rare, if they're not, what to do. I was just reading a case study today about a woman, rereading a case study about a woman who was using voltage-gated calcium channel blockers because she had hypertension. She ended up getting edema in the legs and the feet, which is swelling after use of the voltage-gated calcium channels, and it's a rare side effect. It does not happen to everybody. Most of the studies that I read on them, it does not occur, but you should know about what the rare stuff is too, just in case. But anyway, that person ended up being diagnosed with chronic kidney disease, and then a clinical pharmacologist, clinical pharmacist, basically had to change them from the voltage-gated calcium channel blockers, the calcium channel blockers, right, to an ARB, angiotensin, or an ARB. It's angiotensin 2 receptor blocker, or they changed them. Typically, they change them to an ARB or an ACE inhibitor, or the third one was one more that they also switched them to basically a safer means of reducing medication to reduce their hypertension, so they don't continue to have the swelling in their legs and their feet, and they do not experience chronic kidney disease. I think I put that research article up. I'm not sure. If I didn't, I'll go ahead and put that up as well. Once again, research is for anybody and everybody in the environment. Typically, you want to get a good understanding of it. If it's not your grade level, ask. You know what I mean? I'm probably talking to people that understand this stuff, or that are interested in this stuff, or have a good understanding of the medical sciences, or just interested and want to talk to their doctor about this stuff, right? To see if it's good for them, what the benefits are. So you gotta know how to interpret it as well. In this study, we found that the expression of calcium channels was decreased, and the processes of autophagy were induced by exposure of radio frequency electromagnetic fields. These results suggested that decreased level of calcium channel expression. Once again, that's those gates that are no longer there anymore, right? So we had 5, now there's only, let's say there's only 3. Due to the EMF exposure, the radio frequency EMF exposure, RF-EMF exposure, it reduced the amount of the calcium channel gates. So therefore, the autophagy processes can be activated in the hippocampus of mice. So it basically suggested that the decreased level of calcium channel expression could interfere with the intracellular calcium influx, so basically reducing the rush of the calcium into the cell and protecting the cell. There's no apoptosis or cell death. Therefore, the autophagy processes can be activated in the hippocampus of mice. Further evidence of autophagy induction after RF-EMF exposure is that although hippocampal structures CA1-CA4 and DG were not distinguished in our situation. Due to the difficulty of hippocampal staining, autophagic structures such as autophagosome and autolysosomes were accumulated in hippocampal neurons of the brain of mice exposed to radiofrequency electromagnetic field as a whole. Additionally, we obtained continuously consistent results in randomly cut all area of the hippocampus by TEM, that's the microscopy experiment. Previously, we have reported that 12-week exposure to radiofrequency electromagnetic field activates autophagy in the striatum and hypothalamus in mice brains, whereas 4-week exposure to radiofrequency electromagnetic field does not induce the autophagy pathways in these brain regions in mice. 12 weeks, hippocampus, hypothalamus and the striatum are impacted, right? I'm not sure if it works the same way, that the autophagy protected those neurons as well, but perhaps they did. I don't know. I didn't read that one. But I'm sure in the when I post a link to this in the caption in the video for YouTube, you'll be able to look that up as well. However, we found a 4-week exposure, like I said, leads to strong autophagic responses in the hippocampus of the mice brain. Our results show that autophagy can be triggered in the hippocampus following radiofrequency electromagnetic field exposure as a physiological means of adaptation to cellular stress. So cells feel stressed because of the 835 megahertz radio frequency at that specific power level. The power level is once again 4.0 watts per kilogram, specific absorption rate for 4 weeks. That's the SAR value. However, the processes of autophagy induced by radio frequency electromagnetic field exposure were not observed at hypothalamus in previous study. It is not clear yet why different regions of the brain respond differentially to radio frequency electromagnetic field exposure. We speculated that the structure or function of the hippocampus are more sensitive than hypothalamus to the stress from radio frequency electromagnetic fields. So the hippocampus, their learning and memory center, may just be more responsive and more sensitive to any effects from the external environment, including man-made. The hippocampus belongs to the limbic system and plays important roles in the consolidation of information from memory. But the hypothalamus is also responsible for the regulation of certain metabolic processes. So metabolism also is handled by the hypothalamus, and other activities to link the nervous system to the endocrine system. In addition, the location of brain regions to radiofrequency electromagnetic field exposure or the different tissue configurations could contribute the differential processes. Further, researchers will be required to investigate the differential responses among brain regions. In this study, so there's more work to do, in this study we also found that the apoptosis pathway was inhibited, while autophagy was increased in the hippocampus after 4 weeks exposure of radiofrequency electromagnetic field. It's in figure 3. Radiofrequency electromagnetic field exposure was reported to upregulate the caspase-3 dependent apoptotic pathway in primary cultured rodent brain neurons. Has the citation for that as well. Caspase-3 dependent apoptotic pathway. Similar cellular stressors can cause either autophagy or apoptosis in a cell. The method triggered depends on the threshold of sensitivity to the specific stressor. Both mechanisms are tightly regulated by the inhibition of mediator proteins, BC12 and Beclin-1. Activation of autophagy blocks the apoptosis pathway through inhibition of Bax protein. So thankfully, the autophagy in this particular example blocked or inhibited, right? Stopped the Bax protein. Because if it hadn't, in this example, inhibited the Bax protein, then apoptosis would occur. So that means cell death, cellular death. Anti-apoptotic BCL-2 protein was upregulated, or BC12 protein was upregulated, thereby inhibiting the pro-apoptotic Bax protein in the hippocampus after radiofrequency electromagnetic field exposure. Autophagy is an essential cellular mechanism that processes selective degradation of organelles or unusual protein aggregates. Importantly, dysregulation of autophagy may cause various neurodegenerative diseases. So organelles and protein aggregates is what's involved in the autophagic process. upregulation or degradation or selectively degrading some of those. Importantly, dysregulation of that process, the autophagy process, may cause various neurodegenerative diseases. The analysis of the expression of autophagy-related genes in this study suggests that they may play a crucial role for autophagosomal formation. Activity of endoplasmic reticulum associated class 3, that's P13K, known as VPS34, drives vesicle nucleation by the formation of a multi-complex with Becklin 1/2, which initiates autophagosomal formation. The transmembrane protein ATG9/A recruit lipids to the phagophore. During the autophagy process, LC3 is elevated by cysteine protease, ATG4AB, and undergoes post-translational modifications. A lipidated form of LC3, like microtubule-associated protein 1, light chain 3, is associated with autophagic vesicles. Importantly, a complex of an E3 ubiquitin ligase-like enzyme ATG5 with ATG12 ATG16L1 regulates autophagosomal elongation and is complex. And this complex is essential for LC3-1 combination with phosphatidylethanolamin, it's PE, to form LC3-2 LC3-PE. Eventually, mature autophagosomes that engulfed intracellular organelles fused with lysosome generate autolysosome. Therefore, the level of LC3B2 might reflect the relative amount of autophagosomes in hippocampal neurons. Radiofrequency electromagnetic field exposure significantly increased the protein level of LC3B-2, a protein that is used for autophagy markers as well as represents overall autophagic flux in the hippocampus. It's in figure 3A, you can see that. As shown in figure 3B, exposure of radiofrequency electromagnetic fields resulted in the accumulation of autophagic vesicles, such as autophagosomes and autolysosomes, which might also represent active autophagy in the hippocampus. Thus, radiofrequency electromagnetic field exposure likely induces autophagy in mouse hippocampal neurons. The hippocampus is crucial for its role in the formation of new memories and spatial navigation, and is also involved in learning and emotions. Importantly, dysfunction of the hippocampus may lead to early Alzheimer's disease. The citation there is for 17 and 18. You can look that up in the references. Alzheimer's disease is mainly caused by extracellular amyloid beta deposits, but autophagy could provide neuroprotection through the disruption of amyloid beta aggregates. This study may provide evidence that autophagy offers neuroprotection in the hippocampus in response to radiofrequency electromagnetic field exposure. Moreover, autophagy may prevent neuronal cell death through downregulation of apoptosis, which can lead to neurodegenerative diseases. So those are the neuroprotective effects that were seen, that they hypothesized could protect, could eventually mean that same thing, same logic could be used for people to prevent neuronal cell death through the downregulation of the apoptosis. Because If not, cell death leads to neurodegenerative diseases. Calcium signaling plays a key role in the regulation of neurotransmitter release, as well as cell death. Changes in calcium homeostasis may have significant effects on neurotransmitter release from synaptic terminals. Therefore, radiofrequency electromagnetic field exposure may cause alterations to neurotransmission in the hippocampus, leading to abnormal behavior and or disease states, such as learning impairments, anxiety, and depression. Our study, and that has the citations there, 54 and 55, our previous study demonstrated that radiofrequency electromagnetic field exposure to mice may lead to hyperactivity-like behavior by the generation of demyelination in the cerebral cortical neurons. So I was in their previous study. So that is hyperactivity-like behavior by generation of demyelination in the cerebral cortical neurons. So basically, the neurons not having that protective coating around them anymore, right? So demyelination. So the axons, what sends the signal from one brain or cell neuron, right? The neuron is the cells in the brain, from one cell to another in the brain, were shown in their previous study as a result of radiofrequency electromagnetic field exposure to be demyelinated. That means that the coating around the wires, the telephone poles, are stripped down. So there's no more of that rubber coating around it. So that's what they demonstrated in their previous study. You can look that up. That's citation number 25. However, more detailed studies are necessary to elucidate the scope of impact of radiofrequency electromagnetic fields on the brain. In summary, we found that levels of calcium channels, which play a key role in calcium homeostasis, were decreased in the mouse hippocampus following a 4-week exposure to 835 megahertz radiofrequency electromagnetic fields at 4.0 watts per kilogram is the SAR value, the specific absorption rate, 5 hours per day that occurred. Radiofrequency electromagnetic field exposure likely alters intracellular calcium by decreasing the expression of calcium channels, activating autophagy pathways and suppressing the apoptotic pathway. The expression of genes and proteins related to autophagy were significantly increased. In parallel with the accumulation of autolysosomes in hippocampal neurons, our results suggest that radiofrequency electromagnetic field is acting as a stressor to the central nervous system and causing increases in autophagy and downregulation of apoptosis. So it has the acknowledgments for Dr. HUH, Dr. He, YH, Center for Electron Microscopy Research, Korea Basic Science Institute at Cheongbuk for transmission of electron microscopy. So Dr. Hu basically donated them the microscope. This research was supported by Basic Science Research Program through the National Research Foundation of Korea, NRF, funded by the Ministry of Education. And once again, the title is Exposure to 835 megahertz radiofrequency electromagnetic fields decreases the expression of calcium channels inhibits apoptosis but induces autophagy in the mouse hippocampus. This was written again by, I want to make sure I get all of the authors in here to give them credit for their work. So here it says this article was published in the journal, Korean Journal of Physiology Pharmacology. So the Korean Journal of Physiological Pharmacology in 2018, May of that year. The authors are Ju Hwan Kim, Yu Dong Song, Hung Gung Kim, and Hak Rim Kim. And this is a Creative Commons article. It's open access, so anybody can access it, but you can't use it to make money, right? That's basically what it means. So that is what I have for everyone today. As promised, I will read that article about those effects. Those seem to be positive effects on the surface, but as I've read before, and as the end of that article suggested, that there are other effects such as cortical effects, with the demyelination that they suggested, and other research articles on microwave radiation that I've read before that had that outlined several detrimental effects within the brain as well, including disruptions in the mitochondria. Now that would be the level, I think, would be at 900 megahertz or something like that in that research article. And the absorption rate, the SAR, and the power level, I don't have off the top of my head, but those were also interesting articles that can be replicated and should be replicated to determine where that is happening and if that can inform policy as well. So demyelination doesn't seem like a good thing. Apoptosis doesn't seem like a good thing. Low sperm count doesn't seem like a good thing, as well as the calcium overload does not seem like a good thing, as other articles have pointed out. And also, just off the top of my head, disruptions to the mitochondrial function, the energy centers in the cell, damage to the mitochondria, doesn't seem like a good thing at all. From electromagnetic field, radio frequency, energy, radiation, right? Many of those things do not seem like good things as well. So if companies can still make money, telecommunication companies can adjust their cell phone towers such that it sends enough signal to power the phones, and so that we can still use them and connect people, but without those damaging effects, or have a different way of distributing that energy, or even just protecting the person, focusing on the person to protect them from any damage that might occur. That would probably be very useful. It would probably be very, very beneficial to people as well. So once again, my name is Eric Michael Lloyd. I have a master's in psychology, neuropsychology concentration, and reading to you that article, very interesting article that was published in 2018. I hope that this helps. I hope that people may be interested. Hope that somebody is that there are advocacy groups that are not bullied by lobbyists or anyone else, so that if there are damaging effects to people, few as they may be, those effects can discontinue awareness can be brought forth to many people who have no idea about any of these effects. They can importantly be validated or invalidated if incorrect. Things can be changed, and people could live long, happy, and healthy lives. Because who's going to know about mitochondrial damage or oxidative stress buildup in a cell if they don't look into that, right? It's just, it's not something that I hear every day. Somebody walking around saying, "Oh wow, I got a bunch of oxidative stress occurring in my neurons in my brain. I can feel it." So that's why they measure it, or even let's say DNA damage. So those things can be effectively measured within ethical mice studies, animal studies, rabbit studies. They used for many years to then map that onto what we know about humans and to try to explain what is occurring or what might be occurring in humans that they know are exposed to a certain level of whatever phenomenon it is. In this case, radio frequency electromagnetic fields. So that's how they're able to determine what might be occurring in people and to try to do things to protect the brain, the body, because of that. So hopefully, these things happen if need be, and if it's something that that people can manage without having serious detriments to their health, then nothing will need to change. But that's once again the telecommunications entire mode of making money, right? So I don't want to go into imagining, what arguments they have against it, or what contentions or just or disagreements that they have about what the effects are, or how much power they have over people. But I know collectively, if people are interested in it and can validate this or replicate any of these, even in animal studies, and then very prominent researchers in the field can disseminate this information, then perhaps positive change can be made, alterations to how cell phone towers send the communication, or even radio, public radio, can be made for people in general, to live happier, healthier, have healthier brains, have healthier cells in their body, that don't lead to them having a neurodegenerative disease, Alzheimer's, dementia, Parkinson's, mild cognitive impairment, hyperactivity-like symptoms, or even schizophrenia or schizophrenia-like symptoms. All of these fall within the DSM and the DSM-5, except for the last one was not a neurodegenerative disorder. Schizophrenia is not a neurodegenerative disorder. But anything that could affect the mind through the environment could potentially have a deleterious effect over time on people, on any and every human being. Once again, this is Eric Michael Lloyd. I have a master's in psychology, neuropsychology concentration. Thank you for your time.